methods to interpret signals, as well as improvements in the actual technology, where they interpret the quadruplet nucleotides using De Bruyne graphs. After that, he moved to the Dekker lab, and I found out today that it's actually pronounced Case Dekker. So he moved to the Dekker lab, where they further started working on their own protein sequencer, and on the way discovered a couple of interesting things about DNA process of enzymes. And their approach towards it could be seen as somewhat analogous to using tags, but obviously kind of different. So we're very much looking forward to your talk. I don't know, over to you.
Okay. So yeah, this is gonna be a bit of a radical departure from a lot of the content from the previous talks today, the excellent talks we already saw. So yeah, I'm gonna be talking about developing an entirely new, different technology for sequencing proteins apart from mass spec or the other methods people have mentioned. We get some.
You should only use the lapel mic or that mic.
Yeah. I'll just point it away. Yeah. So I'll start with a little introduction to nanopore sequencing for people who are unfamiliar with it, and then go on to describe the experiments that we did to read peptides at the single molecule level using nanopore sequencing, and then just talk a little bit about outlook and next steps in the development of this new technology. Is it this button? There we go. Okay. Yeah. So the principle of nanopore sequencing, or just nanopore experiments in general, it actually precedes any sort of successful sequencing, just as a way of sort of counting and identifying single molecules. So you have a membrane, very thin. So this is something like a lipid bilayer or a thin crystal like graphene or silicon nitride that you apply an electric current across. And when you apply an electric current, the salt solution that the membrane divides into two wells, the ions are driven through the pore by the voltage. And so you measure this electrical current. It's usually on a scale of picoamps or nanoamps. So you use a very low noise electronic amplifier, but it's really just a basic, it's like an electrophysiology experiment actually, is similar electronics. And so then when something big like DNA goes and blocks this tiny hole, which is about a nanometer across, it blocks the electric current, and the depth of that blockade, is this the laser? No, I don't have a laser. The depth of this blockade here at the bottom, it depends on what the molecule blocking it is. So maybe different content of DNA, if you've got a strand of all T's, you see a different blockage than a strand of all A's. And then eventually, the DNA gets pulled through the pore electrophoretically or electroosmotically, and you return to the open pore current. But the problem is that a lot of the time, generally speaking, analytes go through way too fast. So, you can pull through a many kilobase strand of DNA, and you only see these very, very short little blockages. And so we need a way to slow things down because, we're sampling at kilohertz here. And yeah, and things are going through about 1 million bases a second. So the really clever thing that was sort of simultaneously sort of collaboration between Jens Gundlach's lab, where I did my PhD, starting around this time, 2012, and also Mark Akeson's lab, also pioneers in nanopore research. They collaborated to put together this nanopore called MSPA, which was the first one shown to successfully be able to actually discriminate single nucleotide substitutions in a way that depended on sequence, and then also having a motor enzyme that actually, it acts like it's too big to go through the pore, so it acts like an anchor, like a brake, and it just goes thunk and gets stuck on the top of the pore. And then as it walks along the DNA, it ratchets the DNA, either pulling it up or letting it down through the pore. And so what the DNA bases are in this narrowest part of the pore are changing with every step of the enzyme. And so the sort of simple picture is each of these different transitions here on the right are, one step of the enzyme, the electric current changes because what's blocking the pore changes. And then you can associate these in some way with DNA sequence. But that's sort of idealized, fanciful data. This is what the data actually looks like, this is actually a full read of the Phi-X174 viral genome, which is the shortest known genome, at least at the time that this was collected. So this is a full genome of an organism, and you can just zoom in by a factor of 100 on this top signal that just looks like noise. And you see, oh, wait, that's not just noise. There's a bunch bunch of discrete steps there that correspond to the enzyme ratcheting the DNA through the pore. And then you zoom in by another factor of 100, and you can really see how rich and how clear the signal is. So, these are many times signal to noise jumps because of a movement of one molecule by one nucleotide through the pore, which is pretty cool. And you might also notice there's more than 4 there's more than 4 different levels here, right? You're seeing a lot more than 4 unique states. And that's actually because it's more than just one base that contributes to the current. It's actually a set of about 4 bases that contribute to the current at any one time. But nonetheless, you can still decode the signal and wind up doing quite accurate base calling nowadays. So this has been commercialized, and there's a lot of reasons that it has, it had a lot of purchase in sequencing communities, genomics communities. One is long read length. You can read as long of a piece. There's no real intrinsic limit to the length of the DNA apart from how long you want to sit there and watch the thing read. It's fundamentally a single molecule technique, which is really what makes it interesting, I think, for protein sequencing. So you can detect variations from molecule to molecule. You're not looking at an average of many measurements. And you can use very small sample sizes, like femtomolar or even attomolar concentrations of a given DNA strand can be measured. It's low overhead, it's cheap, it's simple. In principle, you can make one of these devices for 20 bucks in like raw materials. And also the other thing that really makes it useful as a physical or as a protein sequencing tool is that you can directly detect DNA modifications. Or sorry, is that it's like a physical readout. Like, so you can directly detect DNA modifications or alien bases, like non-canonical DNA bases. Because the principle of it is not like based on specific chemistries that attack each individual monomer. It's just you're just physically interrogating this polymer with an electrical signal. So yeah, how are we going to think about sequencing peptides? So I'm going to kind of make the case first that this is a reasonable way to expect to sequence peptides. And so like I mentioned before, you can detect epigenetic markers, and even modifications as small as a single methyl group lead to many times signal to noise changes in the nanopore signal. It's a very nonlinear system. It's not just like the overall volume or size or mass of the thing. It's really, a complex set of interactions between the polymer and the wall of the pore and the ions going through and the hydration shells around everything. So, small changes to the analyte lead to big changes in signal. And even so on the right here, there's a plot that's from Abdelghani Ukalid's group that shows even without this enzyme control scheme. So this is just looking back kind of at these kinds of experiments, looking at just these depths of these little blockades. You can detect single amino acid substitutions in small peptides, like 7 amino acid peptides, and discriminate to some extent what these single substitutions were, just based on those like very narrow few data point spikes. So, one of the first way, to my knowledge, that people really successfully kind of tried to control pulling peptides through the pore, because, of course, peptides, they don't have nice motors like polymerases or helicases that step along them in kind of neat ways. The next best thing is is something like a protease, like an unfoldase, like this thing ClipX. And so Jeff Navala, who's actually has the next talk after me, back when he was with Mark Akeson, did these really cool experiments that kind of pioneered this controlled peptide translocation. Where they got actual distinguishable signals pulling different proteins to the pore. But there are some limitations in the experiment, namely in that this enzyme, unlike a DNA motor, which steps one nucleotide at a time, this thing kind of reaches out and grabs some random, whatever the next byte of peptide chain it can grab, and then it reaches out again. And so you're kind of taking random steps through the peptide, which is really catastrophically bad for sequencing. But it's something that is continuing to be developed. Jeff might talk about it a little bit. But yeah, it's a potential future avenue. But in the meantime, I've been thinking about, my sort of thought was, well, what if instead of having a protein motor, what if we just use the DNA motor, just like we did for nanopore DNA sequencing? And, so the scheme is you have a piece of DNA that you attach terminally to a peptide. So as long as you can do some specific terminal attachment chemistry to a peptide, then you can get one of these DNA handles on, and then use the spatial separation between this constriction, which is what determines the electric current that's actually blocked, and the anchor point of the enzyme, like where the helicase sits, that space is basically the space where you can be walking on the DNA, but reading the protein. And so we're using, the same nanopore I was talking about earlier, MSPA, because of this convenient geometry that separates the enzyme and the constriction. And I'm using this enzyme is HEL308, which is both a very strong motor capable of plowing through just about anything, and it's strong enough that it actually takes half nucleotide steps that you can observe. So which correspond to about single amino acid steps, which is convenient. And you might notice this does put like kind of a length limit on the length of a peptide we can read, which right now is up to about 30 amino acids, but you can engineer the system, make the pore taller, something like, use different enzymes, maybe use a bigger enzyme. All of these things can extend that read length if need be. So, what you'd expect in the end is you get a predicted signal of sequence of these states that, we know what they should be because they're DNA, and we know how the DNA sequence is related to the current, but then we should go on and read a whole bunch of other stuff that we've never seen before. And that's exactly what we see. So on the top is the raw data, and on the bottom, it's just electric current versus time. But on the bottom, what we've done is we've extracted, basically taken all the time domain information out because the durations aren't that important for what's actually in the constriction of the pore. And so this sequence of levels is really what you analyze. And so you can compare the predicted levels from what we know about nanopore DNA sequencing experiments to the measured levels. And we see, yes, it matches the DNA prediction very well, but then we keep stepping and stepping and stepping like we wouldn't. This is just attached to a poly D and E peptide, like a DE heteropolymer peptide, which we chose at first to just make it negatively charged, just like DNA, to keep it electrophoretically, held with force in the pore. And so you actually see three very distinct current signals. So on the left, we looked at three different substitutions of a aspartic acid, a tryptophan, and a glycine. And these three different signals yield, or the three different single amino acid substitutions yield very different signals in single molecule reads. On the right-hand side is just the same data as on the left-hand side, but just with the differences between currents to kind of make clear what's going on. So yeah, that's already pretty exciting, but there's something kind of a little bit non-intuitive, which is, why does the tryptophan actually increase the current? That seems a little bit counterintuitive. You'd think, a bigger molecule blocking the constriction of the pore should drop the current. So we collaborated with some MD theorists who did all atom simulations with the nanopore. And so, they're just simulating kind of half of the MSPA molecule. So, we expect here that the results aren't necessarily going to be exact, like picoampere currents, because they're not measuring for long enough times or the full system, but at least qualitatively, maybe we can reproduce some of what we're seeing. And so what they noticed was some kind of interesting physical behavior in the MD simulations, specifically that tryptophans had this tendency to actually interact with the sidewall of the pore and pull the whole amino acid chain to the side. And so when they're down in the blockage, like blocking the center or just on the bottom side of the constriction, you see a big current drop. And then when they jump up above, then you actually see the current increase, kind of non-intuitively. And so they actually, completely independently of us, this was done as just like a separate experiment, they wound up finding, a certain predicted profile of electric currents that actually qualitatively matches what we actually wind up observing, which is, to my knowledge, the first time that this has really successfully been done with a nanopore system, which is cool. So, yeah, the next question is the obvious question of, like, how accurately can you just take a single read of a single molecule and say which one it is? I mean, do you get it right 90% of the time, 99% of the time? Turns out it's about 90% of the time, a little bit less in our first experiments, which are, comparable to the results for early DNA sequencing experiments before, there's still a lot of development that needs to happen, but that's really not quite good enough. And, if you look at commercial nanopore sequencing devices, single reads, the problem is single reads of single molecules are still pretty low accuracy, but from the, stacked up against other sequencing technologies. But this can improve a lot because almost all of the error in these nanopore reads is random error. It's because of steps that kind of randomly go by a little too fast to resolve, enzyme steps, or the enzyme backstepping, or just noise that's too high so you don't register small steps in the current. But if you measure this over and over and over again, you can average out the random error. And so you can these nanopore sequencers, if you have like 100x coverage, you can get to Q50, which is 5 nines in terms of sequencing accuracy, de novo sequencing accuracy. And there is something very convenient that unlike DNA sequencing, we want this to be a single molecule technology. So we don't want to have to capture 100 copies of the same molecule and read them, because then it's not really single molecule sequencing anymore. But conveniently, there's something I noticed just as I was trying to troubleshoot the experiment and figure out why things weren't working. One day I decided to add a whole bunch of extra helicase. I added, a very high concentration, and I started seeing signals like this, like these very repetitive ion current signals over and over and over again. And this behavior was actually first observed by Art Rand and Mark Akeson's group, and they wound up doing. He wrote his thesis, kind of largely encompassing this idea. But the idea is that when the enzyme is at very high concentrations, it's very likely you have a second enzyme stacked up behind the first one. And then when the first one winds up going down and reaching like the linker or the peptide, it falls off. And that triggers the whole thing to basically be pulled all the way back down. And then you just restart the read from an earlier point. And so we really, using this, just by increasing the helicase concentration or maybe the length of the free DNA, you can get almost indefinite numbers of rereads of a single molecule that are just non-destructive. So you can just read it over and over and over again. And when we do this, we can take different subsets of one of these reads that we know what the true identity of it is. And we can say, okay, well, what if we just use one read? What if we use 2 reads? What if we use 10, 50, et cetera? And we see that our error rate just, as expected, most of this is random error. The error rate just drops off dramatically, exponentially as you increase the number of reads that are used. And so we're able to show that, you can take a single molecule, a single amino acid substitution in a single molecule, and I can tell you what amino acid that was to a very, very high precision if I can just measure 30 reads of the peptide in a row. So next big steps I think are one thing we're looking at now is PTM detection. So that's sort of an immediate low-hanging fruit. These are like big charged groups on the sides of things. And being able to map variants of PTMs, we see one with 2 phosphorylations, 1 phosphorylation and no phosphorylations, and we see enormously different signals. This data is collected by the 2 folks on the screen here at Case Dekker's group. We're working on this now. Also, this question of specifically this arrow labeled conjugate. So, the question of how do you terminally modify one or both ends of a protein? Ideally, somewhat agnostic to what the sequence of the peptide is, or at least in a way where you can calibrate the efficiency of the attachment to what the sequence is. Because and then if we can do this, then we basically are ready to target biological samples as opposed to ones where we've engineered some click linker onto them. And we have chemistries that work for this. So it's just a matter of actually getting them worked into some throughput where we can start with biological cells. So in summary, we have a new protein reading method, that potentially a sequencing method in the future. I think it's pretty promising at least for fingerprinting, PTM identification. If you have a library of signals, of these electrical signals, you can identify which one you're reading with pretty high confidence. But there's a lot of work left. We're trying to get to the point where we're actually reading native biological samples with the technology. We're trying to characterize signals from a lot of different peptides instead of just 3 or 5 or whatever. We need to work on actually maybe some sort of understanding of the physical theory of when you have heterogeneously charged proteins going through. I should have mentioned, these peptides, we have been able to read, they don't have to be all negatively charged DE. You can choose things that have all kinds of different charges, and it works fine. But yeah, understanding physically what to expect and how it should work, also industrializing it, parallelizing it, putting it on arrays of thousands of pores to increase throughput, and finally, figuring out how feasible it is to expect de novo sequencing and what we'll actually have to do to get to that point. There's a lot of challenges in sequencing, 20 amino acids plus PTMs as opposed to just 4 bases from kind of a similar signal. So yeah, just quick thanks, acknowledgements to the Dekker group, where I did all of this work, and various people who've helped in the project, the Aksimentiev lab, who did the MD simulations, the Gundlach lab, who provided us with Health 308 and MSPA, and where I'm actually now working and developing a collaboration with the Dekker group and the Gundlach lab, and also the NVO and the European Commission for funding. So thanks.
Thank you so much for that talk. Do we have any questions? Yes.
It's rather a general question. So to which extent can it replace or complement CMS-based proteomics?
Can it replace what?
To which extent can it complement or replace CMS-based proteomics in the future?
Yeah, I mean, I think it's very much complementary. So I mean, I think that there's the places where a single molecule technology in general can be very useful is when you are talking about extremely heterogeneous samples and samples that are also very low in concentration. Like I mentioned, I mean, we can measure attomolar concentrations of analytes, and the whole thing can be scaled down very, very, very small. So I think there's like specific target applications in that sphere that are the most promising. I have a hard time saying it will replace mass spec, because I think there's obviously many things mass spec is much better suited for than the nanopore experiment. But specifically in the context of just quantifying, identifying peptides that may be in a very mixed sample, a very low concentration sample, I think that's the promising use case for this.
And second question, what is the maximum read length for peptide?
So for it's, this is something that could be subject to further engineering. With the system as it is now, I think the maximum is about 30 amino acids. Most of my reads, that's if you had a 0 length linker, because whatever the linker is between the DNA and the peptide eats into this space between the enzyme and the peptide, or the enzyme and the reading point. And so if you get the linker very, very short, it could be 30 amino acids. Most of my reads are like 25 because of the length of the linker.
Okay, so you don't see any applications for full proteins, for example?
There absolutely are applications. I mean, so yeah, I mean, if you've got proteins that have a lot of repeat regions or, multiplexed type of proteins, then I think, you can't quite tell this. Or if you're trying to look at, for example, correlations between like different PTM sites at different ends of a protein, maybe you'd have trouble with that. But I think most of the question of like identifying a protein or mapping a specific PTM that you're interested in can be handled by judicious choice of where you're digesting your proteins into peptides, like the cut sites, and through and just by looking at whatever these 20 to 30 amino acid fragments are that you look at.
Okay, thank you.
Yeah.
Thanks, amazing stuff. I guess possibilities with the DNA linker could be like super high multiplexed analysis, because you can just change the DNA sequence, read it out, and know what sample that peptide actually came from. Yeah,
we're actually working on a way of doing in vitro translation of like a big library of sample peptides such that they actually get linked to the DNA that coded for them. So then you could quickly generate a big library of sort of like known sequences together with known peptides together with DNA that identify which peptides you're reading, which would be a great training set. In practice, I think it's hard to make sure you're attaching the right DNA to the right protein in a biological sample. So it's not like I could take a cell out of my arm and then if I could label all of the proteins with specific DNAs to that protein, I could just sequence the proteins or the DNAs. That'd be easier.
Yeah. So, I mean, I'm completely new to this, so I was really fascinated hearing such a nice intro. But what would it take to do whole protein sequencing then?
Whole, you mean like
Like
de novo sequencing and
To get rid of the peptide step.
the peptide step, sorry.
So cutting the protein into peptide is something we
Yeah, you know, I
have
mean,
to
I
do,
think
but-
for end-to-end protein sequencing, I think you know, further development of Jeff's strategy is maybe a little bit more promising if you need end-to-end sequencing. But then the caveat is you really have to figure out how do you get a reliable stepping motor that walks purely on proteins, right? Because this is in principle capable of reading a full end-to-end length of a protein.
Oh, amazing stuff. Thank you.
Thanks.
Hi, thank you. I wonder, with your specific aspartate and glutamate example, did you look for, or can you look for isoaspartic acid formation?
Yeah, that's an interesting question. I have not looked for that, but you know, I think in principle, it's been shown that with in fact, there was recently a paper, I forget who it was, somebody showed that with MSPA, actually the same nanopore, if you dangle two just chiral variants of the same exact protein in, you get different signals of the same exact amino acid in, or the same chemical ligand in. You wind up getting different signals just from chiral variants of the same molecule. And so, yeah, like isoforms, definitely, I think you could tell the difference between. So far, I mean, in my experience in nanopore sequencing, I've never seen two physically distinct molecules pulled through a nanopore that didn't at least yield slightly different currents if you average long enough.
Interesting.
Because your error in the actual electric current measurement is really just kind of like your averaging time. It's your error in the mean of each of these steps, which, you know, if you're reading for, you can read just longer steps and you'll get a more precise measurement of what that mean current is.
I guess I have a follow-up question. Have you looked at other substrates like lipids or glycospecies or glycopeptides?
I have not. Other people are working on that, actually. I think that going into looking at glycans is something people are actually just starting to do. I just saw some talks, like unpublished work kind of about people starting to explore pulling different glycans through. People have pulled glycopeptides through, you know, and measured different variants of glycosylation on peptides with nanopore sequencing. And that's capable of distinguishing those. I mean, something as big and floppy as a glycan you'd expect should give a pretty big signal, I guess. But we're actually working on, in addition to the phosphorylation experiments, we're also looking at glycosylations of MHC peptides. Yeah.
Like linear or fresh?
Right now, just linear, simple ones. In fact, we're actually thinking about we might chop them down or digest them in some way to make them easier to go through. So it's not so much that you could necessarily detect every different variant of a sugar. That's a pretty ambitious idea, I think. But you could maybe detect whether there was a glycosylation there or not.